So Christophe will talk to us about the standardized pipeline for the analysis of mass spectrometry-based single-cell proteomics data. So I will pin you so that you are okay. I will close.
Okay, and you can see my screen now, right? Okay, perfect. Thanks. Yeah. So hello everyone. Today I'm going to present you the progress on my work. I'm Christophe. I'm part of Laurent's lab. Laurent that just presented. And my objective, so the title of my work is to indeed develop a new framework to allow and to standardize the analysis of single-cell proteomics data. I'll maybe start with a high-level idea of the project. So my work is doing bioinformatics, and what is bioinformatics? This is actually understanding biology from data, and this is through a whole process. And this process includes first identifying a research question and having a sample of interest that we process through some assays. Those assays, from them we can collect data, and from the data that we analyze, we can get knowledge. There are several things we can do. So there are several types of measures. We can think about sequencing, probing, microscopy, simulations, but there are of course others. And we can think at looking at the data, well, the biological question at different levels. And we can think about epigenomics, genomics, transcriptomics, proteomics, and many others again. My work is focused on analyzing sequencing data in proteomics. Of course, those assays, the sample we analyze, usually we analyze it as a whole. And this is known as bulk assays. And from bulk omics, what they do, they generate a single observation actually for a highly complex sample. And we could even think like even what seems to us homogeneous samples, we could think about cell lines. Those are actually also heterogeneous because of internal variation. I see two main issues with bulk assays is that, first of all, suppose we have a heterogeneous population with two different cell types. Well, having only a single observation for those populations, well, we miss actually this heterogeneity. So we think we are analyzing one cell type or one process, but actually there are several. There are two different kinds. There's also something else if we're interested, for instance, in differentiation, where processes are more like continuous and yeah, it's not black and white. Having a single observation for those, we completely miss this continuity. And so therefore, in the recent years, well, single-cell omics have been developed. And here, single-cell omics is generating a single observation for each cell separately. And this is very powerful because this also allows to unlock new analytical tools. However, it's very nice, but still there are some challenges. And again, I could categorize three main challenges. So first, technical challenges. So we have single cells, and usually it's not one or two single cells, it's more like thousands or even millions of single cells. Well, this requires automation. And so engineering, we are working with minute samples. So imagine how much information or how much there are very minute amounts of sample within a single cell. And of course, if we are analyzing a lot of cells, we want the cost per cell to be minimum, because otherwise it's just unaffordable and hence unusable. There are also computational challenges. Many more samples means also big data. There's also, because of minute samples, there's dropouts. And dropouts refers to the fact that when the levels are so low that sometimes we're just not able to analyze it. And you can see it on the bottom left, on the bottom right, sorry. You can see that for some cells, we have no information. Then you also have important batch effects that needs to be carefully corrected for. And then finally, we have conceptual challenges. And here I think this is very interesting and more philosophical, let's say, or more food for thought, is with single cell data, we can think about what is actually a cell type. it's normal, so clear cut. What is a cell type? Where do we put borders? Where do we put thresholds? Also, what is biological relevant? Before this, we could think about cell cycle. We want to synchronize cells because otherwise we're measuring only cell cycle, and this is not biologically relevant. But actually, now we can deconvolute this with single cell. This becomes very interesting. So yeah, those are challenges that makes this whole field to me very interesting, and that's why I thought let's get into it. Another thing is, so here what I showed, the field of single cell is actually is getting mature, especially for the single cell transcriptomics. However, transcriptomics is a good proxy for proteomics, but it's actually the proteins that are doing the function. So having more information about protein means having more understanding, or a different kind of understanding on how the biology works. And actually what has been shown is that even though on the global scale, RNA levels follow very well protein levels. So if we look at different genes across different samples, we can see there is a good correlation. But if we look at a particular gene across different tissues, we can see this correlation becomes much worse. So for instance, even if you look at gene 3, we get anti-correlation. That's the first thing we can think about. And then another thing about proteomics is that the protein content, the amount of protein copies within a cell is much more important, is much higher than from transcripts. And this has a very important implication statistically, is that we are more, it's easier to sample the true amount of proteins. Because with RNA, sometimes, well, if you have very small copy numbers, it can just happen that you, by the PCR, just do not catch up that RNA. And so this is just statistically, it's not even technical. And so because of those two reasons, single cell proteomics is becoming, is very interesting and is becoming more advanced. And so indeed, because in recent years, and actually, so here I show you some figures about this year, recently, we were able to, the lab from Nikolai Slavov was able to quantify over 1,000 proteins in over 1,000 single cells. And so this is done through mass spectrometry. So the idea of mass spectrometry is you have a cell, you split it up, you extract the proteins, and you split the proteins in peptides. Those peptides are then separated through liquid chromatography. And every peptide afterwards, we get a mass print or a spectra that allow us to both identify and quantify that peptide. And so there are two main strategies. Either we do it label-free for more accurate quantification, or we can do it multiplex to increase the throughput. The issue here, the thing is that this is very recent. And so we have a lot of development on the single cell transcriptomics. So a lot of methods are available. On the other side, we start to have very interesting data about single cell proteomics, but we don't have the methods that allow this analysis. And so the idea is to combine those two fields in a single infrastructure, computational infrastructure. And so we came up with this idea. So we call this SCP infrastructure. The idea is we use a particular class in R. So those are, what I will show are bioconductor classes. So we combine the Q features that is ideal for analyzing mass spectrometry data. We combine it with single cell experiment objects that are ideal, that allow us, that are an interface to single cell methods, single cell, for the moment, mainly transcriptomic methods, but that can be applied to single cell proteomics. And in this idea, based on this framework, we developed two packages, scpdata. So this is a package that disseminates datasets, the currently published single cell proteomics datasets. But then we also have SCP that allows to do streamline analysis and already perform single cell proteomics data analysis. I give you here a small example of the use of SCP. So on the left, you can see this is a framework. This is probably not the ideal framework, but this is something that was used already to analyze currently the data. And on the right is our implementation of this pipeline using SCP. And you can see, well, I mean, you don't need to be expert in R, but from the few functions and if you understand a bit mass spectrometry, you understand already what it does. So the idea is that this software should be accessible with minimal requirements in terms of programming. Okay, so we have infrastructure, we have some packages, but is it actually useful? And to answer that question, we reproduced the scope 2 dataset. So that's the one dataset that's current state of the art. That's the one dataset that's quantified over 1,000 cells. And by applying our tool, we could see that numerically. So on bottom left, we can see that the differences we observe are very minimal. And on the right, we can see also, so the idea is to reconstruct we have the different spectra, and what we want to do is reconstruct the data towards the protein level. So have protein information. But this reconstruction means aggregating things. And if we look at the end, the proteins that come out of scope 2 after filtering or after different processing steps are the same as the one we obtain. So with more than 97% overlap. So this is very good, indicating that indeed our methods is able to reproduce things. But you might say, yeah, okay, you still see differences. So maybe something completely changed. So in order to check that, we performed some kind of custom PCA. And to see in the data, what are the relationships between the different cells? How are they dispersed? Maybe the small differences make big shifts in the data, but this is not the case, as you can see here. On the left, we have the original. This is a published in bioRxiv, but it's still published figure. And on the right, that's our reconstruction. And you can see also there are small differences. Those small differences are not important. I mean, in the sense that we can still neatly distinguish between the two cell types. So in this case, macrophages in blue and monocytes in red. So this is one example I gave you of one replication. So you can see the replication is almost perfect. I mean, that's not the thing. The interesting part here is that actually we were able to reanalyze this data using the same methodology. But doing this allowed us also to understand new metrics for single-cell proteomics that were described in the paper. But it also highlighted issues and possible improvement in the analysis. And this, I think, and that's to come up pick up with Laurent's presentation, reproducibility also promotes actually analysis improvements. We did the same thing on another published data set, but the lack of good documentation didn't allow us to neatly reproduce it, but still to neatly replicate it, but we still could reproduce the methods. With SCP, well, I mean, this is just the beginning of a journey. There are still a lot of challenges. So namely batch effects. So you can see batch effects on the identification. So on the features we find, depending on the batch, well, we find different things. So how to keep up, to take that into account, but also batch effects in terms of amplitude of measurements, as you can see on the right. There is a lot of missingness. This is something that is typical of single cell, but this is something we should carefully take that into account. They can be biologically, so kind of expected missingness, but also technical missingness that we want to remove. And this requires specific models. And namely one model that I think is interesting and that I will dig into, but I think it's worth coming up, is the hurdle model. Basically, this model is able at the same time to look at differences between expression. So we could do differential expression gene analysis, but at the same time, we can also look at differential detection probabilities. So meaning, do we see this gene in one cell type, maybe not in the others? Is this statistically different? So this is a model very interesting. Okay, as a take-home message, what I just want to say to you as a conclusion is that single-cell proteomics is an emerging but very promising field. We have developed a computational infrastructure to formalize this data analysis. We were able to apply this infrastructure to 2 published works. And finally, there are still a lot of exciting challenges that need to be solved. So I'd like to thank Laurent, my promoter, but I would also like to thank you for your attention. And then I'll make a little advertisement. The Bioconductor is giving a conference in 2 weeks where I will also present this work. So I'll be happy to see you there. There's still time to register for it. Thank you very much.
Thank you very much, Christophe. Yes, please feel free to pose any questions you have. We have some time for a few questions. I have one, although I'm not an expert in mass spectrometry, so maybe what I say it's not applicable. Is there a limitation on the type of data that this pipeline could be used, or how it could be improved? For example, I don't know. It's just
No, I think there's still a lot of improvements that can be done. I mean, this, as I said, is very early. There's still a lot of research to be done on this.
Yes.
I'm for the moment very inspired by the single-cell transcriptomics or single-cell RNA-seq analyses, which there are already like amazing work done on that side. And I think definitely we could get inspired. And some methods I could even like, for instance, trajectory analysis, if you're a little bit in single cell, that it's a type of analysis where you can see how cells differentiate based on single cell data. Well, some of it has already been applied to single cell proteomics. So yeah, definitely this is something that could be done, but there are other some challenges that are more specific to proteomics and so require the dedicated analytical methods. And this is something I'll be working on in the coming year or years.
Thank you. And then we have a question from Laura, actually. What were your biggest challenges in trying to reproduce the SCP results?
Yeah, for me, the biggest challenge is, how to say it, to wrap this whole method in some clear, well, if I take, oops, wait, you can still see my screen, right?
Yes.
Yeah, so basically taking existing, and the existing analysis, so with the whole R code was provided. So for this is very good, and I could actually running their code exactly reproduce what they did. So on the reproducibility side, they did their job, but on the usability and on user friendliness, that was not the case, and it took me a lot of time to actually understand what the code was doing, and then also convert it in a, like, let's say, standardized and easy to understand, easy to read way in this package, and make it user friendly. Yeah.
Okay. And maybe a last question from Chris. Are there already textbook examples where single-cell proteomics answered questions which were not possible with single-cell transcriptomics? If you know.
Yeah, well, textbook, that's maybe still a little bit early, but I think so that one example I gave with the macrophages and monocytes, well, I mean, this whole experiment was made to show indeed that this could be applied to actually textbook questions, let's say. And in this case, the question was, they know monocytes are differentiating into macrophages, and you know there are, well, if you know a bit about macrophages, there are 2 types. There's the M1-like and M2-like. And apparently things that were not clear yet is this M1, M2 something that is black or white, or is it something that is continuous? And from this data, so I don't show the analysis of that, but from this data, it seems that this is a continuum. So that's, you can see a continuum between M1 and M2. Yeah, so that's, for instance, one example that could be done. Differentiation could be another example, clearly, but that's not done yet. I mean, not clearly.
Okay, we have like 30 seconds, if it's possible. Okay. A last question. Can you comment on strategies to deal with batch effects?
Yeah, that's a good question. Yeah, so Well, in 30 seconds, it's hard. I mean, I would say that there's like classical, you can do some regressing out the batch effect. I think it's more interesting to put that directly in the model. So for instance, if you do differential analysis, now include the batch effect as a covariate or with some special distribution maybe, or like using mixed models and using batch as one of the levels in the mixed models. Mm-hmm. So there are different strategies. I think I would say more, I would be more interested into modeling this directly and do some analysis, some inference based on that. Okay. But yeah, there are different ways, and still we don't know what's the best way.
Okay, thank you.